Hey there, biology. Mrs. Cozen here, and we are about to do notes about meiosis or meiosis. You can call it whichever one you want. I usually say meiosis. Quick little review of your last set of notes, though, about mitosis, okay? So make sure that you can answer these 4 questions about mitosis, okay? Make sure you can do those 4 things. Okay, what is cancer? The uncontrolled growth of cells. The cells don't turn off. Steps in mitosis. Interphase, prophase, metaphase, anaphase, telophase, cytokinesis. And interphase and cytokinesis are not technically a part of mitosis, but we're going to stick them in there. How many cells are created by cell division or mitosis? You get 2 cells at the end. They're called daughter cells, and they are identical. Okay? If you guys had any trouble with any one of those questions, I want you to go back and read those notes again. Okay, so this next fill in the blank notes about meiosis. You guys are going to see words like gamete, haploid, diploid, the word meiosis, crossing over, and the principle of independent assortment. Remember, you don't have to write this part. We'll do these in the notes. Okay. Meiosis itself is the process by which one gamete divides into 4 daughter cells with half the chromosomes. So first, let's talk about what a gamete is. A gamete is a cell that is used for reproduction. It's a cell that makes babies. So it's, in humans, that's sperm and egg cells. So this meiosis only applies to sperm and egg cells, okay? Did you write that down somewhere? Only applies to sperm and egg cells. Mitosis, however, the one you learned about yesterday, that applies to body cells, so every other cell in your body. Only the cells that are used for reproduction that are going to pass information down to the next generation go through this meiosis, okay? I want you guys to circle 4 daughter cells with half the chromosomes, okay? So if you have a cell that goes through mitosis, and let's say the original cell had 10 chromosomes, both of the new cells will have 10 chromosomes. But when you have a cell that goes through meiosis, if it had 10 chromosomes at the beginning, at the end, each one is going to have 5 chromosomes, okay? So make sure you guys also that you've written down what gametes are. Those are sex cells. Meiosis only in sex cells, only in cells that go through reproduction. Okay. And here's the process of meiosis just mapped out. So you guys can see we start with 1 cell and we end with 4 instead of 2. Okay. And you can also see we had like 4 pieces of genetic information here at the top, and then we only have 2 here at the bottom. So you get half the information. Which is really important, because when we're going through reproduction, sexual reproduction, you can only have each parent pass half the information, half the genetic information, to get an offspring that has all the genetic information, okay? So for you guys, you have half your genetic information from your mom and half from your dad. So in each one of those gametes, those sex cells, it had half of the information for you. We wouldn't want to have all of mom's information and all of dad's information. That would be too many chromosomes for you. Okay. Now, vocabulary to describe those concepts. The first one is haploid. Think half. Okay, I want you to write the word half somewhere next to this. Haploid means half the amount of DNA. So in humans, that means that in sperm and egg cells, there are 23 chromosomes. Okay. In all your other cells that go through mitosis, like in your skin cells, there's 46 chromosomes. But in your sperm and egg cells, there's 23. Okay? That's because sex cells only pass down half of the genetic information to the offspring. Okay? So if the word haploid means half, that applies to meiosis. The word diploid applies to mitosis. Okay, so somewhere next to diploid, I want you to write the word mitosis. That means double the DNA, 2 sets of chromosomes. So this is what we call body cells. Okay, so I want you to write body cells somewhere near this. These are the cells that we're not using for reproduction, so they need all the directions. Okay, so these are the ones that are going to make, when they go through mitosis, they're going to make identical cells at the end. Okay, so diploid, body cells, identical, haploid, sex cells, and they're not identical. Okay, so let's go through phases of meiosis. The phases sound exactly the same. You just go through them twice. So we have interphase. Remember, that's the longest phase. It's where the cell's growing and copying all of its different parts to get ready for division. Okay. In meiosis, we only go through interphase one time. So add that next to this, please, okay? Interphase only once. So even though we're going to go through 2 phases of cell division, we're only going to do this first phase one time. Okay? And we only do it one time because remember we end up with half the chromosomes. So if we did interphase, if we did all that copying twice, we'd end up with 4 cells that have double chromosomes, and that's not what we want here. Excuse me. Okay. First phase, prophase 1. I want you guys to put a big circle around the 1. So this is the first time that meiosis goes through prophase. You guys can see if you're going through prophase 1, you still only have 1 cell. Oh, sorry guys. Oh no! No, go back! So back to prophase 1, you only have 1 cell that's dividing into 2. Okay? First thing there says homologous chromosomes pair up to form a tetrad. These things are a tetrad. So take, or tetrad, you might hear it called. Take this word and point over here. You guys can see this is enough chromosomes for us to eventually make those 4 cells. Okay? And I want you guys to circle the word homologous. So that means the same chromosomes. So like these are all the copies of chromosome 1, and these are all the copies of chromosome 2. So chromosome 1 might have all the information for your eye color or your hair color. And so all that information is lined up together. Crossing over occurs. Crossing over is really important, so put a big star or something next to that. Crossing over is where these get shuffled around. So if all of this is all the information for chromosome 1, and let's say that's for your hair color and your eye color, that's all the genes that you possibly have for hair color. And they get shuffled around like a deck of cards. So that when we get our 4 cells at the end, not every cell gets the same information. Okay, so this thing right here, this little concept, it's the thing that makes every single one of you in this room different. Okay, look at the people around you. They are different because of this crossing over concept. Okay, just like in mitosis, we start to lose that nuclear membrane and we start to see those chromosomes. So the chromatin started to wind around those histone proteins. So we have a better package now for division. Metaphase 1, just like metaphase in mitosis, our chromosomes line up on the middle of along the equator, and then the spindle fiber is going to grab them. Difference, if you guys will look back at metaphase in mitosis, we've got twice the number of chromosomes here. Because remember, I'm going to end up with 4 cells at the end. So in mitosis, I only had this many. In meiosis, I have quite a few more. And remember, metaphase 1 means 1 cell dividing into 2. Anaphase 1, same as in mitosis. We just have more chromosomes in that 1 cell. Spindle fibers grabbing onto those chromosomes and separating them, moving them to opposite poles. And then last but not least, telophase and cytokinesis 1. So this is where we get down to 2 daughter cells. Are we done yet? No, because in meiosis, we've got to make 4 daughter cells. So we're going to go right back to prophase again, and we're going to divide the 2 into 4. So prophase 2, you guys can see this is supposed to represent 2 cells. Okay, so we've got 2 separate cells. 2, we already have 2 cells and we're dividing into 4. Okay, again, spindle starts to form, and the centrioles, remember those are the things that are like the reel on the fishing rod. They're gonna start to move towards the poles, and we're gonna start to get that nucleus to break down again. Metaphase 2, very similar to metaphase 1. If you guys will go back and look at the picture of metaphase 1, though, we have fewer chromosomes here because we're going to break it down into 4 cells. Okay? Now it says they're not in homologous pairs. That means that our chromosomes are not in pairs that perfectly match. Remember how we went through that crossing over in prophase 1? Now I might have all the information for like chromosome 1 here, but let's say chromosome 1 has the information for your hair color. If you had some genes for blonde hair and some for brown hair, maybe some of these are blonde and some are brown, okay? So these don't perfectly match this time. We don't want them to match because you wouldn't want every sperm and egg to be the same. That would be boring. Anaphase 2, same as anaphase 1, but now I've got 2 cells going through it. Compare the pictures, okay? You guys should be looking at that paper back and forth. Look at anaphase 1, anaphase 2. Now our chromosomes start to split in half. Now I've got these chromosomes are going to be in one cell. These are going to be in another. Almost there. And finally, telophase and cytokinesis 2. We form a nucleus, cytoplasm divides, and we have 4 haploid daughter cells. I want you guys to circle 4 haploid daughter cells. Okay, so this is a big difference between mitosis and meiosis. We've got 4 cells in meiosis. You only have 2 in mitosis. They're haploid, half the DNA. Mitosis is diploid, 2 sets of DNA. Okay. All these cells are different. In mitosis, they were identical. All right, so just a quick recap. You guys don't need to rewrite every one of these, but just a comparison between meiosis and mitosis again. Meiosis, you only do interphase 1 time. If you did not write that, I want you to go back to interphase and I want you to write that. You only do it once. Then you go through and you divide 1 cell into 2, then you divide 2 cells into 4. All right, you get 4 genetically different cells with half the number of chromosomes. Here is the concept of crossing over, and this is why those cells are not the same at the end. Okay? Homologous chromosomes exchange some parts during meiosis. This happens during prophase 1, so somewhere around here I want you guys to write prophase 1. And this is the concept that makes, like I said, every single one of you different. It's what makes every living thing on the planet different from its brothers and sisters or its parents. During prophase 1, all those chromosomes get shuffled around so that we don't necessarily have everything exactly the same in our sex cells at the end, okay? Which is great. It gives us genetic differences, and then when we get to talking about evolution, this is going to be really important. This is how we get, crossing over is how we get new traits. It's how things evolve and change over time. It's very important. I think I have one more for you. Principle of independent assortment. This is the idea that when we are making sex cells during meiosis, the genes for different traits stay separate. So when I say that crossing over is happening, it doesn't mean all the chromosomes in the whole cell are mixing with each other. It means that all of the copies of chromosome 1 are mixing together. So all the possible genes that you have for eye color are mixing together, and then they randomly get assigned to the cells at the end. So if you have blue eyes, but your parents had brown eyes, that means you most likely have a gene for blue eyes and a gene for brown eyes. Don't worry about what color your eyes are yet. We'll talk about that second semester. But when you make those gametes, when you guys are creating those sperm and egg cells, some of them will get the blue eye gene, some will get the brown eye gene, and it's not attached to anything else. Okay, so they stay separate from each other, and they stay separate from your other traits. So like your eye color is not necessarily attached to your hair color. They're separate. Okay, crossing over happens different for those. Your eye color isn't attached to your height. Okay, not everyone with brown eyes is tall, or not everyone with blue eyes is short. So this is just the idea that different traits stay separate. Okay, different traits stay separate from each other. We'll do this later, and you guys are done. Nice work. Thank you.